Again, welcome everybody to Paleo Talks, being brought to you by the Center of Excellence in Paleontology at East Tennessee State University, which oversees the Gray Fossil Site and Museum. And all of us on here, except Sam, are part of that. And Josh is on here in particular today because Josh is also a rodent expert. And so, coming in with Sam's topic today. And Sam, as we get started, if you could just go ahead and show your first slide. And then what I want you to do is go ahead and get into your background and how you got into paleontology in the first place. Just sort of tell us that story of your paleontology, maybe where you went to school as well, and what got you into working on things like rodents.
Great, I'll be happy to do that. So I grew up actually in East Tennessee. So we were talking about this a little bit ago, but I grew up in Knoxville, and I went to the University of Tennessee for my undergrad. And when I started college, I thought I was going to be a conservation biologist. That was where I was really interested, was in, conservation issues and questions of extinction and, human impact on species and that kind of thing. And so I was going through with that assumption in mind, got involved in evolutionary biology, was doing undergraduate research in that and that kind of thing. And my first senior year, I took a paleobiology class in the earth sciences department, just out of curiosity, and just got super jazzed about paleontology. And so I took 5 years to finish undergrad because I double majored in earth sciences. And I went from there to graduate school at University of California, Berkeley, working with Tony Barnosky. And he was really interested. He was one of the kind of early adopters of conservation paleobiology. So looking at the paleo record as a way of understanding, current conservation issues and thinking about the processes that might shape, the ongoing biodiversity crisis. And so I worked with him to kind of learn how to address those kinds of problems. And since then, I've really been trying to pursue research that would help us understand where biodiversity comes from and how, the diversity of mammals evolved and what ecological and evolutionary processes shape them going forward.
So Sam, along those lines, this has been really exciting to watch this growth of conservation, sort of paleobiology. Do you think we're at a point where we're starting to see actual policy developed as we're making discoveries?
I think we're not. It's not my perception that we've quite gotten there yet. I think we have gotten to a point where evolutionary biologists are recognizing the value that paleontology brings to answering evolutionary questions. And we've started to see some work done in the realm of evolutionary biology that's trying to use the evolutionary histories to inform conservation strategies a bit. But I don't know that we're quite getting to the point where that integration has fully taken place, where findings from the paleo record are really influencing policy. But I think we're not far from it, because I really do see those conversations kind of headed in that direction. I see, people really starting to realize the value of the paleo record for understanding where we've been and where we're headed.
Great, thank you. We'll go ahead and start working your way through the presentation. Thank you.
All right, great. So today I'm going to talk about a tiny little piece of research that has a little bit to do with conservation paleobiology. But it's kind of part of my long game, trying to figure out what shapes the evolution of ecology in fossil mammals. And I'm going to talk about a group of animals, the Applodontiids, that I've been working on since I was in graduate school. When I started graduate school, my graduate advisor handed me some undescribed fossils and said, go figure out what these are and write a description of them. And they turned out to be Applodontiids. And that project led me into this whole diversity of fossil rodents that have really kind of formed the core of my research since then.
Sam, right now we're seeing the presenter view.
Oh, nuts.
With that again.
My apologies. Let me switch. I will fix it. Okay. Is that better?
Not yet.
Uh-oh. Okay, we'll stop the slideshow a sec. And
It
I'll
might
try.
help to start the slideshow first and then share the window of the full slideshow
Got it.
instead of sharing the PowerPoint. That happened to me the other day.
I
the
will
whole
try that.
screen. Share your whole other screen.
Okay, we'll bring my slide back up. And let's see. No, I don't know if that's gonna, yeah, there we go. Do this here. Sorry for the chaos. We'll just
No
see if
worries.
we can get this to behave itself. And that. Aha. Too many options. Okay, how's that?
Perfect.
There
Awesome.
we go. Worth the wait.
Okay, so now you can admire my cute little Applodontia here. So I'm going to talk to you a bit about evolution in this group, and I'm going to explain why a fiddly little bit of taxonomy for a group of rodents that is not all that widespread might be of broader interest than you'd think. So this picture here that you see on the slide is the one living representative of an entire family of rodents. So this is Applodontia rufa, known as mountain beavers, even though they're neither beavers, nor do they particularly live in the mountains for the most part. And they're endemic to the Northwest, which makes them rather relevant for me at the University of Oregon. Not that I could have known that when I started working on these guys. But one of the things that's interesting about them is that you'll see them referred to as primitive rodents. And I'll explain to you first why people call them primitive. Let's see if we can get this to, there we go. All right, my slide's actually gonna advance now. So here's the thing. Among rodents, rodents are an immensely diverse group. There's something like 1,500 living species of rodents, which is about 40% of the diversity of mammals, right? And so in looking at the sort of taxonomy, the evolutionary history of rodents, a lot of the early efforts to organize them focused on the morphology of the jaw muscles. So the masseter muscles in particular, this is the muscle that closes your jaw when you tense your jaw or clench your teeth. This is the one that that you feel that connects the skull, the zygomatic arch here, your cheekbone to the lower jaw. And in rodents, something goes on with these jaw muscles that you don't see in anything else, which is that they move the attachment of the jaw muscles forward on the skull over evolutionary time. And it's happened several times in rodents. It's clearly, there's sort of an evolutionary push to increase this forward leverage of the masseter muscle, which almost certainly has something to do with, the other thing that makes rodents rodents, which are these giant, ever-growing incisors that they use to do all kinds of things that rodents do. And so there's 4 categories of rodents into which all the rodents have been placed based on the way that these jaw muscles attach to the skull. And the figures that you see here illustrate what the morphology is that puts this together. So almost all mammals are what we would call protrogomorphous. And this is the kind of, starting point. The masseter muscle attaches to the underside of the cheekbone of the zygomatic arch here, and then down on the lateral side of the lower jaw. And the 3 different ways that rodents change the masseter attachment are called cyoromorphous, myomorphous, and hystricomorphous. And basically, myomorphous is like cyoromorphous plus hystricomorphous put together. So cyoromorphs, you can see this is the lateral masseter, the outside part of that masseter muscle. They move that attachment out onto the side of the snout, on the side of the rostrum here. And you can see hystricomorphous rodents do not do that with the lateral masseter. They actually have rather a reduced little lateral masseter. But hystricomorphous rodents have this giant infraorbital foramen in the zygomatic arch that reflects the fact that the medial masseter, the interior-most part of the masseter, actually has run up onto the front of the snout through the infraorbital foramen, so through this hole in the cheekbone. So either way, you attach the muscle further forward on the jaw, you get better leverage up at the incisors. But those two approaches to moving this muscle forward evolved separately, right? And then myomorphs do both, right? So they've both got the medial masseter run out through the infraorbital foramen, and they've run the lateral masseter up forward towards the rostrum. So three different ways that have evolved to change from this protrogomorphous state among rodents. And we see, big diverse groups that have each of these things. The hystricomorphous condition is really characteristic of South American rodents, for example, things like capybaras and all that kind of fun business. Sauromorphous, as you might guess from the name, really common in squirrels. Myomorph condition really common in mice and rats and things like that. But several of these evolve multiple times. The thing is, this protrogomorphous condition is not all that common, which isn't surprising when you see all these changes happening, right? Like, kind of expect that the ones that didn't do anything might not diversify as much. And in fact, if we look at the evolutionary relationships among rodents, this is what we find, right? So the colors here show the different jaw muscle types, and it may be a little hard to read, it's kind of small. But myomorphs are these little orange dots. Hystricomorphs are the ones with the little black dots. Sauromorphs have the blue. And then protrogomorphs are green, okay? And this is an evolutionary reconstruction of the relationships among rodents. And what you can see is, here's a bunch of hystricomorphs, right? Here are a bunch of myomorphs, these are mice. Here are some sauromorph things that are little squirrely relatives, right? As well as some others that have evolved this hystricomorphy. But you'll see there's only 2 green dots, so only 2 extant species in this analysis here. There's a lot more rodents than this, but there aren't a lot more protrogomorphs. Only 2 extant protrogomorphs, okay? One of them here, this is naked mole rats, and they've reduced all kinds of things in the front of the skull. And you can see really clearly in evolutionary time, they've kind of lost the hystricomorph condition, okay? They clearly evolved from something that was a hystricomorph. So Applodontia here is the only thing that is primitively protrogomorph among the rodents that are alive today. And so you can see why, with all this emphasis on jaw morphology in how rodents are related to one another, people might think that Applodontia is a primitive rodent, okay? But the question we have to ask ourselves is what the retention of this primitive jaw musculature means for the evolution of skull morphology and of the rodent as a whole, right? Can we call it primitive just because it retains this primitive jaw morphology, okay? So this is the question that I wanted to ask. And I'll show you the short answer to that question, but there's sort of a longer answer that I want to get at too, right? So the short answer is the rest of it is not primitive, okay? So on the left here, you see an image of Applodontia. This is a skull. You have a ventral view, a view up on the palate here, and you have a lateral view of the same skull. And just for comparison, the skull next to it here is of Parames, which is an Eocene rodent, a common Eocene rodent, has morphology a lot like what early rodents have, okay? And you can see several things about Applodontia that are really not at all primitive and that are quite derived relative to Parames. So you can see the shape of the skull. Parames has this narrow back of the skull, just like most rodents that we see. And Applodontia has this really broad back of the cranium here, right? The ear bones here are really modified. You can see they're pretty typical in Parames, but they're really, really modified here in Applodontia. And so the zygomatic arch here, the cheekbone, is maybe the only thing that's all that primitive about Applodontia, right? The other thing to take note of here is these teeth look pretty strange. They're sort of a little peg of dentin surrounded by some enamel. And you can see the roots of the teeth go way up into the skull here. Here's a lower jaw of Applodontia just to compare to the tooth row here in Parames. You can see the teeth are really different from where they come from too, right? This derived morphology in the teeth is really striking.
Yeah, that changed in looking at the occlusal surface, the chewing surface of those teeth, that is so bizarre.
They're really weird. It looks like a little Salvation Army shield there in the uppers. And the lowers look basically like a letter B. But you can see that in Parames, they have low crown teeth like we do. In Applodontia here, this is the base of the tooth crown. You can see the little bump on the side of the jaw here, where this tooth goes all the way down into the jaw. And there's all this modification of the jaw to make room for that, okay? So these are not primitive. So great, there's your answer, right? Like, just because the jaw morphology is primitive doesn't mean that everything else is, okay? But so why? When does it happen? When does it change? And what makes this happen, right? So this is a question I wanted to ask. So this is a phylogeny I built quite a long time ago of all described species of Applodontians. There's a lot of them in this clade, and you can see there's also a lot of diversity here. So the color coding refers to crown height of the teeth. These high crown teeth are clearly a really big part of the question. The black down here are brachydont things, low crown things like Parames. And as the colors get warmer, crown height gets higher, and they go all the way from this low crowned sort of squirrel-like tooth morphology to modern Applodontia rufa here, which is the one remaining species in this group, the one that's still around, which has what we call hypsilodont cheek teeth, which means their teeth never quit growing. They grow throughout life. And so they have essentially infinite amount of tooth crown to chew their food, okay? And it turns out that the changes in the skull morphology that I pointed out are kind of tied to this change in crown height. So this skull on the right here is of a thing called meniscemes. It's one of these green lines down here. I believe it's actually this one right here, if you care. And meniscemes is just starting to see a little bit of an increase in crown height, and you can see that the skull shape is still very much like Parames was. So you can see pretty typical auditory bullae as you see in other mammals and so forth. So the change in hypsidony seems to be pretty well connected to the rest of these changes in skull morphology, which isn't really surprising. You change the shape of your teeth, you're gonna have to remodel your skull all around it. And you can also see that the jaw morphology in meniscemes was pretty consistent with squirrels and all the other rodents that you see. And so the weirdness in Applodontia, what we want to do is really figure out where hypsidony changes. And the other thing we kind of want to look at is body size. You can see on here that the changes in body size occur kind of interestingly at points when crown height increases as well. So I wanted to focus in on this group here, the Applodontianae, which are just the clade most closely related to the extant Applodontia, and really look at this evolution of crown height, because you can see they go through a dramatic change in crown height relative to other Applodontians. And here's a sense of that, right? So they start from something that's mesodont, like this guy here, Niglerodon. Many of them are hypsidont, have high crowns. You can see the tooth root, and this is a worn crown, so there would have been a little bit more of it. And of course, the modern ones are hypsilodont like this. They have an open tooth root and a really tall tooth crown, right? So you can imagine cramming that into the skull is not trivial. The other thing you'll see is that there's not a huge number of species here. So it's not hugely diverse taxonomically. And that's kind of an interesting phenomenon. But one of the things I realized was that part of the reason why this taxonomic diversity was so low was that there were actually a large number of undescribed Applodontians in museum collections that nobody wanted to deal with. And we could talk about why nobody else wanted to publish the Applodontian species that were lying around, but there you have it. So the paper that resulted from this basically started with a simple species description and grew into this analysis of Hipadani evolution and Applodontians. The fossil that kicked it off was this one. This is skull here you see on the left side, Lyodontia dalei. This was a group of fossils that were originally described in a dissertation by Michael Moria in 1981. I was 4 at the time. And I found this skull in the collection at UCMP in Berkeley. And I asked the collections manager, like, wow, this is an amazing skull. Look at this thing. And it's not a published thing. How is it not a published thing? And she's like, well, this guy did a dissertation on it, but then he didn't stay in academia. And so he didn't really get around to publishing it. I was like, but it's important. This is like a big thing. You know, this is a really cool fossil. And part of why it's really cool is that it's one of the earliest things in this clade, the Applodontianae, right? So its age is late Hemingfordian, which is the North American land mammal age for about 16, 17 million years ago. And this site is interesting because it's also one of the first occurrences of elephant relatives of proboscideans in North America. And so, you know, there's a lot of interesting things going on in the fauna, but it also means it's been dated really confidently. And so we really know that this is one of the earliest Applodontians out there. And there's also a lot of material. So here is a whole series of tooth rows from the same site, except for this orange guy at the bottom here. So we have like more than a dozen partial skulls and jaws of this material. And it's really completely known. So you can see this complete skull here. We have several complete jaws from which to describe the fossil. It has a couple of features that most of you may not care all that much about, but it has these little infoldings of the enamel in the back of the tooth. This posterior reentrant on the M3s, both the uppers and the lowers, you can see. And this is an interesting feature because in this group, in Lyodontia, this genus, you're not supposed to have that. That's supposed to be a characteristic of the more basal members of the clade. And so it's part of the reason I got excited about this, because I was like, oh man, we should have this published, right? This is important because it shows us a transition out of some of the more basal Tardontia type Lyodontia or Applodontians into some of the more derived higher crowned ones. I got really interested in this question of crown height, but as you might have noticed, it's a little, it's tricky to know exactly how much tooth crown there's going to be if the roots aren't closed yet, because you can still grow more tooth crown. And so I started looking at kind of the proportion of the specimens in an assemblage with closed roots, because that tells me something about how long that root stayed open, right? So if you sort of randomly sample from that, then you'll expect that if they stay open longer, then you're going to get fewer with closed roots. And so this one, most of the specimens had closed roots, which is interesting. Tells me it's hypsodont, but definitely not a hypsilodont, not an ever-growing tooth here. And then just sort of an aside, I found some more specimens of this from a place in Montana, and that becomes interesting a little bit later on. And then I went to the Burke Museum, and I found another undescribed Lyodontia. So this is one from a place called the Deep River Formation, which is why this is Lyodontia bathypotamus, which is a taxonomic name that made me really happy. And again, we've got a really good sample of this thing. Again, it's from that middle Miocene interval. And again, it shows us sort of lower crown height. It shows us these transitional fossils in the Lyodontia clade.
Sam, they have a very restricted geographical distribution today. What was it like in the past?
Well, so one of the things you can see here is this guy from the Deep River Formation is from Montana. And one of the things I've been finding is that, you know, the early, so like in Oligocene time, these things were pretty widespread. We find Applodontiids in Nebraska, in the Great Plains and so forth, in South Dakota. By this time, by the middle Miocene, these guys are mostly like Montana and then the Northwest, as well as Nevada. So kind of Great Basin, Northwest, and then into the Northern Rocky Mountains. And where I'm going is that when we get later in time, This is the third new species I described in this study. This is a latest Miocene, so about 5 million years old. This guy is from Oregon. This is the first occurrence of Applodontia, the genus, in the fossil record. And it's a little one. It looks like its other buddies. But part of what we're running into is, I think, part of why we don't know as much about these things and where they were. This is an immense microfauna, and there's 2 specimens of this taxon, the whole thing. So you're looking at them here. There's a nice jaw, fortunately, and then there's this one little isolated upper molar that we have, right? So by the time we get to the end of the Miocene, I think these guys are limited to the northwest from the look of things. Some interesting morphology. There's some changes in the jaw muscles or in the jaw shape that I'm not going to have time to talk about. So I took those 3 new species and threw them into basically a comprehensive description and phylogenetic analysis of the Applodontianae. I realized the only way I was going to figure out what they all meant was to look at the whole stinking clade. And so there are a lot of them. Here are some more. I kept thinking I was just going to do a little description paper, and then it was going to be a slightly bigger description paper, and then all of a sudden it was a revision of the entire subfamily. I took all the characters I could find on the teeth and ran a big phylogenetic analysis of every single Applodontian I could find. There's about 12 species of them in the record anywhere, along with 4 of the species that might have something to do with them to be outgroups. And in short, we actually got a surprising amount of resolution, even though these things are high-crowned and they eat all their characters. That's one of the challenges for phylogeny and things like this, is that they chew all the shape off their teeth. And so given that rodents are known mostly from jaws and teeth, it can be awfully hard to reconstruct phylogeny with something like this.
How does this phylogeny based on morphology compare to what people might be getting in genetics work with these critters?
So the nasty, unpleasant thing about this is that Applodontia rufa right here is the only one you have any hope of getting any genetics from, because all these guys are Miocene and older. And so many of them are extinct. And it's one of those clades where it's like, oh man, wouldn't we like to know, right? And all we've got is morphology, and it's morphology that's tricky to work with because they're mostly known from isolated teeth. You got to deal with tooth wear as the characters change. You got to hope you can find some juveniles that haven't chewed all the characters off their teeth yet, right? So it's tricky stuff. But I did at least manage to get pretty good resolution. And it's fairly concordant with our taxonomic expectations, right? So based on what was there before. The unfortunate bit is that it's very clear that this has been kind of a series of taxa that have evolved one after another in this sort of comb-like pattern. And so all of the genera that people had described before were based on, primitive characters. And so the genera are all paraphyletic, which annoys the phylogenetic taxonomists a bit, but c'est la vie, right? So looking at the question of Hipadani evolution that I started this with, if we look at this sort of percent closed roots as our proxy for Hipadani, here's what that phylogeny tells us about how that evolves. Essentially, for the taxa where we know this change happens quite quickly. So we go from Tardontia and Pseudaplodon, all of which are rooted. If you find them, if it's not like a half-formed baby tooth, it has closed roots. You get into these 2 Lyodontia that I described. Lyodontia bathypotamus mostly has closed roots, but a few don't. So you can see that kind of increase in crown height. Lyodontia dalei is like, a quarter of them don't have closed roots, right? And then Lyodontia alexandrae, still we're operating in the space of like a couple million years. Lyodontia alexandrae, half of them have open roots. And everything that you find after that, it's Hypsilodon. Almost all of the tooth roots are open. Occasionally you find a really old individual with closed roots, but it's super rare. So all of the action in Hypadani evolution, everything that made Applodontians what they are, happened in the space of a couple of million years in the middle Miocene. So that's pretty cool. Interestingly, it also happened in places that are really volcanically active. You can see Tardontia here made it into the later Miocene and is not hypsodont, but it doesn't occur in Oregon, where volcanoes were really lighting off big time. So Oregon is a big part of this story, which is kind of fun.
So is this a grit-related change?
I think so. And there's been some other studies, some of the South American studies have suggested that what was going on in South American ungulates, where they get extreme hypsodonty early, might have had something to do with volcanism as well. So we have reason to suspect that, you know, hypsodonty evolution in these guys might be a grit-related thing. The other thing that I pointed out earlier that I just kind of wanted to circle back to a little bit is body size. So part of this picture is also thinking about when they get big, right? Because Applodontia is a pretty good sized rodent. It's a loaf of bread sized rodent, right? And we know that rodents don't really start there. They start more sort of squirrely sized, right? And it turns out that that is a really recent phenomenon here. So even when we look back at Applodontia micra, that 5 million years ago, it's still in that squirrel sized range for the most part, right? We're sort of in the half a kilogram of rodent size. And so this is another bit of relatively rapid ecological evolution. Just in the last 5 million years or so, Applodontia rufa has gone over a kilogram and has gotten into the really big size and the ecological characters that come with that. So the last point I wanted to make when I look at this evolutionary history is the geographic one that Blaine kind of started us on earlier, which is where all of this is happening. And I apologize, these stars are a little easy to lose on the map here. But what you can see, if we look at these, the orange stars are the group out of which Applodontians came. And so those you can see range over the Northwest and into the Rocky Mountains for sure. The record at the right time in some of these more southerly areas is not so good. There wasn't much of Nevada in the late Oligocene. Most of Nevada arose since the late Oligocene. But the less hypsodont members of this group, Tardontia, for example, occur really commonly down here in Nevada. And then the more hypsodont ones occur in the more northerly sites. And then here's Applodontia, our hypsilodont, most hypsodont group. And for a while there, I thought this was geographic partitioning until I found the site Juntura, which has in the late Miocene, it has a Tardontia in it and no Lyodontia. And it's a really screen-washed, like really well screen-washed fauna. So seriously, Lyodontia wasn't there. So the simple story I wanted to tell about how, the things that were living in the not so volcanic area were all lower crowned than the things that were living in the volcanic area is wrong. So there's clearly some kind of sophistication to this geographic pattern that that isn't as simple as the story we might like to tell, which, you know, surprise, evolution of ecology is complicated. But it's interesting that this clade seems to have been limited to kind of the Northwest, the Great Basin, and the Northern Rockies through much of its history, which isn't true of some of their relatives. So, you know, the mylogalids, the you know, horned gophers or whatever they call them, that are the sister clade to this group, get all over the West and all the way down into Florida through the Miocene. And these guys just don't. They're not as widespread. Okay. So what do we know based on describing a few species and running a phylogeny of Applodontians? These guys, the clade leading to the extant Applodontia represents a huge amount of morphological diversity. There's a huge range of evolutionary change in the group. They've been relatively low diversity through time, which is kind of interesting because the one that made it through is out of this kind of low richness, low diversification rate clade. They did have quite a bit of morphological diversity in the middle Miocene, right around that Hemingfordian Barstovian boundary, around 16 million years ago or so. They also underwent rapid hypsodonty evolution at that time. So in the sort of Barstovian they seem to have done a lot to change the crown height in their teeth, and then undergone a really rapid body size evolution during the Pleistocene, so in about 5 million years ago, into into recent time. And then, you know, my take home from the question that started this thing is, as a result of looking at all this morphological diversity in the skull and in the jaws and in the teeth, it's probably not a great idea to use extant Applodontia rufa as an exemplar of a primitive rodent, because most of the morphology is actually quite derived. The fact that they haven't changed the jaw muscle attachment on their skull does not make them a primitive rodent. And so using these as your, like, you know, this is what your early rodents looked like is rather misleading, because they're dramatically morphologically and ecologically different from the place where they came from.
And I think that's the case with most organisms that we look at today or in the past, is that it's always a combination of derived and so-called primitive characteristics, and it really does them a disservice to refer to the organism itself as primitive.
Yeah, absolutely. And these are definitely a, you know, case study in this. So
right, yeah.
So that's what I have to say. You know, it's a short story, but I'm happy to take questions from anybody who has things to say.
Excellent. Well, we have some questions coming in. Let's go to any that we might have from our panel. Josh or Chris?
While we wait for the panel to jump in, I want to remind everybody watching that now's the time to send in your questions on Facebook or through our other social media sites, and we'll start reading them out here in just a bit.
One thing I was going to ask is, do you think that other aspects of the anatomy of these things are actually a good analog for a primitive rodent? Things like the ear and the brain case, are those indicating features that you might have had in early members of this group, while the skull and jaws and teeth are really heavily modified for a particular lifestyle?
So that's a great question. I can only answer part of it. The ear is a bad idea. So I don't know if you noticed this in the skull I put up. I'll go back through here. If you look at the otic capsule here, so the ear region of the skull, this is really quite different from your basic mammalian ear. So this elongated, you know, elongated tube leading to the actual external ear here. And actually, if you look at the internal structure of the bulla, there's some really interesting things going on in the ancestors of these guys. They modify the bulla to put all these little tiny bony partitions inside of the bulla. And it looks a bit like the inside of the auditory bulla in kangaroo rats and things like that. It's not as enlarged, but it has an internal structure rather like that. I don't think we have a great sense of what that means for their ear function, but I do think that given all that modification, expecting the internal ear, for example, to look anything like a basal rodent is a bad idea, right? You don't have to go back that far back before it's not that it's not quite so much like that. So even in let's see, oops, sorry, there we go. Even in this guy Meniscemys doesn't have that elongated middle ear there, but it does have the septe inside the bulla. The ones that don't, if you get back into the things at the base of the tree here, these blue lineages, Alamys, they have just a few septae in the bulla, so it's a little bit modified. But unfortunately, we don't have a whole skull really much of Alamys. I think there's one that I've seen because they're kind of fragile and they get smooshed. And so once you get far enough back that you don't see these modifications of the basocranium, you're in things that you don't have skulls for. So I don't know what the brain case is like, and I don't know if there's a lot of modification there. I know there was someone who was doing some CT work scanning skulls, including Applodontia, and I think it'll be interesting to see where that gets us, but I think you'd want to scan, some meniskemes and some of these Lyodontia and kind of try to get a sense of what's going on in the brain case to know whether or not looking at Applodontia's brain case was going to be indicative of what's going on.
I think that was Ornella Bertrand who was doing that.
That's right. Yes, thank you. I think it names.
So the skulls don't tend to show up very often in a lot of members of this clade. It's interesting because the other group you show there on the left, the mylogolids, they all have that really interesting skull morphology. They're the horned gophers in some cases, but there's a lot of skulls of those. So do you think the fact that you've got a really good fossil record of skulls of those, but not this group, is reflecting some ecological differences in how they use the environment and what they did?
Absolutely. I mean, mylogalids, of course, are burrowing, and Applodontians are also, but I think we have a pretty good idea that mylogalids, that these horned gophers and their relatives, are much more highly adapted for burrowing than Applodontians ever get. And they're also modifying the way that they dig. So mylogalids are headlift diggers, and so they use a different digging approach, right? So Applodontians dig with their forelimbs, and you don't have to modify the skull as much for that. You still have the wide, flat skull that's adapted for packing the roof of the burrow, like in almost every burrowing mammal. But mylogalids are actually using their face to excavate their holes. And so you get all kinds of modifications to the back of the skull and the front of the skull and everything else, because it's become a spade. And so yeah, I mean, there's some really interesting things going on in there. And that's the subject of a variety of other studies. I know Jonathan Khaled and I have talked a lot about ways to figure out what's going on in the mylogalids, right?
Let's go over to the Facebook questions for a second and circle back to Chris. David?
Sure thing. I'll start with this one from Charlie. How does the jaw morphology of mylogalids compare with that of Applodontia?
That's an interesting line of inquiry, actually. So Applodontia, and I realized I don't have a great picture to show you, but Applodontia turns the angle of the lower jaw under, and I think it's probably related to some modifications to the pterygoid musculature, which is another of the muscles that sort of stabilizes the jaw during chewing. But it makes them look really strange relative to other rodents. It's one of those things that people have gotten really struck by. And you can see meniskemes here has pretty much a squirrely, flat little lower jaw that doesn't have that modification to the back of the jaw. Mylogalids are somewhere in between. They do have a little bit of expansion of the attachment for the pterygoid, but not nearly as much as in Applodontians. The other thing that we see in mylogalids that we do see in Applodontia is the modification for accommodating those big, tall, high-crowned teeth, because you can see mylogalids also evolve really high-crowned teeth, and they start to run into crowding issues. So as you put that crown down into the jaw in reserve, it runs up against the lower incisor. It does this in the top too, so you get the top teeth often curve out so they can wrap around the brain case, so you don't drive the roots of your teeth up into your brain. Seems like a bad idea, right? And so you see a lot of modifications all around that to accommodate all that tooth crown that they're storing both in the lower jaw and in the skull.
All right, I'll go ahead and ask one more. Well, where am I? Here we go. Our friend Grant has asked, has any isotopic work been done on these taxa? And same question for microware. And if so, do isotopes and microware track with the evolution of Hypsodonty?
So I just got the isotopic data back this week, and I haven't had a chance to process it yet, but tune in soon. We've run a little bit of isotopic work. We're mostly looking at oxygen isotopes because one of the problems with these guys is that they don't really occur in a whole lot of C4 ecosystems. And actually had a paper about a decade ago where I established that it looks like there's a negative correlation between the abundance of Applodontoids and the proportion of C4 in the ecosystem. So they don't seem to like C4 ecosystems at all, which makes stable isotopic work harder. But now that we've gotten to where we can tell sort of open C3 from closed C3 environments, we're hoping to do a little bit more in this. But right now we've been looking at oxygen isotopes as a proxy for water use because modern Applodontia is also weird in their kidney structure. They don't concentrate their urine at all. They're basically constantly drinking and living in relatively moist habitats today. That works great. But one of our questions was like, how could they be in Eastern Oregon 5 million years ago when it was really quite dry in the Pendleton area? And so we're trying to look at oxygen isotopes to get a sense of that. Currently our carbon numbers don't look so great. So I'm not sure if we're going to get anywhere with that. As to microware, we haven't done it yet, but we're thinking about it, I guess. Rodent microware is a little tricky. It's a lot easier to do in big things. The scaling doesn't work so great, and rodent microware data seems to be really noisy in my experience. So if we can overcome that, I'll be happy to see what microware has to say about it.
Right,
That
Chris,
was
are you
actually
waiting
my
on
question,
Dex?
Sam. Well, I'll jump on there because Grant actually was, he and I must be on the same wavelength because I was thinking, you know, the biogeography of these things, this range keeps constricting through time. Are we looking at an ecological niche that, I mean, clearly it's contracting and they're not adapting to new conditions. And, you know, you're mentioning, well, they don't do C4. Do you think that that's kind of why? I mean, are we seeing regionally, are we seeing kind of the rise of C4 at that time? And that's why perhaps this is happening?
Yeah, I mean, this has been my speculation. Like I say, this is a paper that I wrote a little bit ago, and correlation does not necessarily imply causation. But the strongest correlate I could find for kind of the decline of this entire lineage here, right, of these, 100+ species of rodents that, as you can see, was really quite diverse in the Oligocene. It seems like, the thing they couldn't keep up with was the spread of C4 grasslands. They really just, when that gets going, these guys kind of start to peter out. And so that's our best guess, is that the C4 grasslands, either because of the physical environment they represented or because of the ecological environment, right? The plants that were there, maybe these guys don't like to eat them. It's hard to say because the extant ones are just weird. So they eat bracken fern and they eat, like all the garbage in the understory in the Coast Range. They eat bark. Part of why they call them mountain beavers is that they will chew down saplings as well. And they're a pest species in the timber stands in the Coast Range here, because they'll murder all the little tiny saplings, and the loggers hate them. So I mean, the modern ones are eating stuff that like probably was not what sustained them in the past, right? But they certainly aren't eating straight up grass, right? And they're not eating C4 basically at all today, right? And so it's an open question, and it's something that I think does need treatment with things like isotopes and microware to really figure out how far back these dietary patterns go.
I was going to say, an interesting thing about this is the same time that some of these groups were dwindling is the time that things like ground squirrels and voles were taking off.
Yeah.
And those things, some of them, like voles, do well on eating grasses, but not all ground squirrels and marmots and things like that are really eating a lot of grass. And it's interesting that you've got some things really taking off at this time and other things vanishing.
Yeah. Well, and when I was looking at this, as part of my dissertation, I was trying to figure out, and my guess was, oh, they were outcompeted, right? We all want to fall back on that competition argument. I was like, that was what I expected to find, that some other group of burrowing mammals had like horned in on their territory. Haha, it's funny because some of them are horned. And like had taken over that niche. And if you look at the patterns of diversity in the things that are ecologically most similar to them, it doesn't look like that's what's going on. So I think it's actually the ecological shift that the voles and the ground squirrels liked better, rather than competition. And I know you weren't necessarily implying one thing or another. I'm just sort of, I think it's worth pointing out that, we often have this guess that when you see one clade increase in diversity and abundance and another one decline, you're like, competition, right? And actually, when you drill down, it really doesn't look like that's what's going on. The patterns of geographic occurrence, the patterns of diversity, and the things that are ecologically most similar just don't match up in this case, and it doesn't seem to be that way. So I thought that was interesting.
And I kind of think that's what I had in mind, is this kind of changes in the environments and habitats, and some things like it and some don't. It's kind of like horses and camels really did well at the same time period, and other things like Oreodonts vanished.
Right,
and
it's not
rhinos
that they
also
outcompeted the Oreodonts. It's just that Oreodonts didn't like it so good once it got all nice and open, right?
Yeah.
All right, David, do you have any others?
Sure do. Here's a question from Elizabeth from Hidalgo, Mexico, who asks, so how would you explain that those primitive characteristics persist to the present in extant species of Applodontia?
Great question. Why didn't they change their jaw morphology, right? Like, this is the $64 question. Everything else was doing it, right? Why did they get left behind? And there's a trite evolutionary biologist answer, which is that evolution doesn't always give you what you want, right? So maybe they would have liked to have, jaw muscles that moved forward on their rostrum, and they just didn't get it. But there's the less trite answer, that is, maybe this group was unusual enough relative to other rodents in the way that they did things, that demand that seems to have been there for a lot of rodent clades just doesn't seem to have driven change in these guys, right? So the jaw muscles, for whatever reason, seem to have been adequate for what they needed in this case. The need for that anterior leverage doesn't seem to have been as great in these guys. I don't know that there's anything in the ecology or the morphology that particularly justifies that. It's not as though these things don't use their incisors or use them in a way that is unusual relative to other rodents. They do a lot of the same kinds of things that other rodents do. They haven't gotten into any of the more extreme, like incisor behavioral modifications. They're not one of the ones that's digging with their incisors. So there are groups of rodents that do that. But if they're chewing down saplings, that's definitely putting some real forces on their incisors, and it seems that their jaw muscles are adequate to handle that. So it's a great question why they don't seem to have made that change, and whether that's just a result of never getting lucky evolutionarily, right? Never getting that change that would move the jaw muscle attachments forward, or if it's a matter that the selection just wasn't strong enough to change that, is an open question.
Well, speaking of comparing rodents, Greg McDonald has asked, a wide occiput is common in many burrowing rodents that use the head for digging. Could its presence in Applodontia reflect a burrowing adaptation?
It probably does, but it's not. So the rodents that use the skull itself as a digging tool, not only have a wide occiput, they also tend to tilt the occiput. If you have the skull right side up, and so this is dorsal and this is ventral, they tend to tilt it forward a bit to accommodate bigger neck muscles. because the leverage it takes to excavate with your head is quite large. And so if you look at things like at Spalax, which is a mole rat, if you look at bamboo rats, which are head lift diggers, if you look at golden moles, which are non-rodent head lift diggers, all of them have an occipital plate that tilts anteriorly. And mylogalids do too. Mylogalids almost all have a forward tilted occipital plate. It gets more so as they get bigger and so forth, which is exactly what you'd expect in a head lift digger. In Applodontia, the occipital plate is totally vertical. And furthermore, it isn't very tall from the foramen magnum, so there's not a lot of leverage there for the muscles that lift the head up. There's more leverage for the muscles that pull the head side to side. So that's interesting, and I don't think we have a great sense of what they're doing evolutionarily that justifies the broadening, the widening of the skull without making it any taller. So still working on it, but man, the skull morphology is weird in these guys.
Excellent. Well, we are just about at the end of our time. So if anybody else on our panel has any last questions or comments, now's the great time for it.
I was just going to mention those kinds of jaw muscles, putting some of the muscles forward on the face. It's not just rodents that do that. There are South American ungulates, these noda ungulates that do that. There's other groups that this is a common feature of herbivores that basically want to chew better.
Mm-hmm, mm-hmm. Yeah, it's an unusual, it's not super common in mammals, and there are plenty of herbivores that don't do it. But it's interesting that rodents almost all do it, right?
Mm-hmm.
That this is their thing. And I think it's probably a reflection of the use that the incisors are put to.
All right. Well, thank you so much, Sam, for coming on the show today. That was great.
Absolutely. Enjoyed being here. Thanks for inviting me.
You bet. We've got a lot more rodents to cover, thousands it seems like. So maybe we'll just have rodents, from here on out.
Rodents for days.
All right.
All right. And before we wrap up, Blaine, should we say that next week's Paleo Talks is going to be a bit of a special one?
It is going to be a special one. So because it is Darwin Day. So we're going to be celebrating Darwin Day on Darwin Day. And we're going to have Dr. Thomas Holtz with us. And he's going to be talking about Darwin and his role in looking at fossils.
Should be a lot of fun. I'm excited for it.
Yeah, if you don't know who Thomas is, he's a well-known dinosaur paleontologist, but also a bit of a historian in Darwin as well. All right.
Great.
Thanks again, Sam.
Thanks everyone for joining us. We'll see you next time.